Welcome to Chemisode on proteins. This is part 2 for proteins. So we're going to look at how proteins can be used and how they get their structure. So let's look at proteins. Alright, so far in proteins you've basically looked at what they look like in the chemical form, you've looked at the molecules. Now we're going to look at the overall structure of what proteins are. Proteins are massive, long chains of amino acids. Okay, so what they are is these long chains of amino acids, and what happens once you get starting to get really long chains is they start to kind of fold around and they start to get really interesting structures. There's 3 parts to a protein structure. There's the primary structure, the secondary structure, and the tertiary structure. And we're going to look at how these structures form. First of all, the most basic of them all is your primary structure. This is basically just simply the order of the amino acids. So it's like the name, when we're writing the name, the order of the amino acids. This is how the structure is. It is just a straight order of those amino acids. Basically saying we first of all have an alanine, then the whatever the rest of them might be. So that's the order, the primary structure. The bonding involved in the primary structure is simply the peptide linkage which is linking them together. So the covalent bond is the peptide linkage which is holding the primary structure together, and that's a very strong linkage. It takes a lot of effort to break that. It takes a hydrolysis reaction to break the primary structure. The secondary structure of a protein is looks like the coiling or the helic structure. So the staircase structure, so you know DNA is a double helix. Basically take one of those, is how it coils around into like a spiral, is the secondary structure of a protein. It gets this structure by the backbone C double bond to O and the NH groups. So those 2 groups which are in your amino acids, so the C double bond to O group there is from your carboxyl group, and your NH is obviously from your amino group. When you form a peptide linkage, these guys kind of come into play and they start to have hydrogen bonding, pushing them together or pulling them together, and it kind of coils your protein into its secondary structure, into its staircase structure, the spiral structure. So the secondary structure is hydrogen bonding within the peptide linkages in the backbone of our protein. The last, probably the most important structure, is the tertiary structure. The tertiary structure is the overall 3D shape of our molecule. So as you can imagine, once, first of all, the protein gets coiled up into a helix structure, after that it starts to fold and pleat and shape itself into the overall 3D shape of the molecule. What makes this overall 3D shape is the Z side chains. There are a few different things in the Z side chains which can make things fold around and interact with each other. The first thing is the ionic interaction between so the amine bases and the carboxyl groups which are in the side chain. No, those ionic interactions are when they're in different mediums. So when they're in the zwitterion area, you get these ionic interactions between the side chains. The second thing is covalent linkages, the disulfide linkages. So what can happen in your cysteine or your CYS protein, it has a sulfur in it. What can happen if you get 2 of these in your protein? These can actually join up and form a covalent link or a disulfide link. So that will only occur in those special proteins. The next type of force can be a dispersion force within a side chain. So if your side chain is nonpolar, you're going to get a dispersion force holding it together or moving it around. And the next one, which basically is obvious, is hydrogen bonds within your side chains. If you have an alcohol group, or if you have a hydroxyl group, or if you have an amino group in your side chain, you're going to have hydrogen bonds, and that's going to start to make your folding, your pleating, your overall 3D shape. So there's the structure of the proteins, your primary structure, just simply the order held in place by strong covalent peptide linkages. Your secondary structure, which is your coiling into a spiral, and that is hydrogen bonding within between two peptide linkages. And your tertiary structure there, which is your overall folding and pleating, which is interactions from your Z side chain. So the side chains in your molecule interacting and making your tertiary structure happen. Now we're going to look at the uses of proteins. Now we're going to focus on two uses, those being the highlighted and underlined ones in your notes there, the enzyme and the medicine. But I'll quickly just explain where they can be used in terms of the other ones here as well. So let's have a look at the uses for proteins. So the first use of proteins here is in terms of structure. Structure in proteins basically means proteins are used in your different areas of your body which are used for structure, such as your skin, such as your bones, such as your muscles are made of proteins. So these, in terms of structure, that's where proteins lie, in those areas of your body. So your skin is made of protein, your muscles made of protein, and so on and so forth like that. Things that create your structure. Small proteins, or relatively medium-sized proteins, are used as hormones, chemical messengers in your body. So you know what hormones are. You know that when your body wants to send a signal around your to another area of your body, it can do it two ways. It can do it by nerves, which is an electrical impulse, or it can do it by using a hormone, which is a chemical signal, which is released by a gland in your body and is picked up by another part of your body where it can do something as well. So hormones are proteins, and they are another way that you're used in your thing there. Protection. Skin is a type of protection, really. It's a layering, so that's where it's used. But also protection in terms of pheromones and different things like that, and toxins in your body. So toxins in other animals as well, especially when some things are toxic, the toxins that they release are generally made up of proteins. So they can be used in the protection areas of your body as well. So toxins are a form of protection in a way. Enzymes we'll look at in detail. Medicine we're going to look at in detail. And a last resort for your body after it's gone through all your fat stores and after it's gone through all your carbohydrate stores is use for energy. So if you get right down to your skin and bone area, your body will start to eat your proteins and it'll start to try and get energy from your proteins. But that's a very, very last resort. So we don't really use proteins for energy unless we're absolutely starving and we've been out in the desert for a long, long time. Anyway, let's have a look at these two things here, enzymes and medicines. First up is enzymes. Enzymes are basically biological catalysts. They are things that speed up reactions. The way they work is an enzyme is a large protein which has been folded into a three-dimensional structure. So your tertiary structure is very important with enzymes. It is very important because it has a little site where a molecule can attach to, and that's how it will catalyze the reaction. Two words you need to understand is enzyme and substrate. Your substrate is the molecule that you are catalyzing the reaction of. For instance, this example here, this diagram, shows maltose and the enzyme which is going to break down maltose. Maltose in this is acting as our substrate. So our substrate is our reactant. And our enzyme is our catalyst. We have an area which the substrate fits really nicely. So we have what is known as the active site, is a shape, a specially designed active site, a shape in the molecule where that the substrate can fit really nicely. This is your active site. The reason it fits nicely is because enzyme activity is a kind of a lock and key. It will catalyze only one reaction. It is very specific for the substrate. The method that happens is we first of all have the enzyme and substrate as separate in diagram 1. In the second diagram, we have the enzyme substrate complex where they are joined together, where this substrate forms probably hydrogen bonds or dispersion forces on the active site. And lastly, what happens after it joins with the active site, it breaks up into the products that we're forming. So obviously we're doing a hydrolysis with maltose, and the enzyme is catalyzing the hydrolysis of maltose in this diagram here, this example. Let's move on to see how enzymes can be affected by different things. I think that's what we got. Enzyme denaturation or denaturation. Enzymes will stop functioning if their shape is affected. You can imagine that if we stuff up this shape here, what happens is the active site no longer has this perfect lock and key. If we can imagine it being a lock and key, someone is trying to break into your house. If they don't have the right key for that lock, it won't. If the key doesn't match the lock, it won't open. So if we change what the lock looks like, we won't be able to catalyze that reaction. Say you've gone out somewhere where your parents didn't want you to go, for instance. What they do, if they get really, really angry, is they change the locks so you can't come home. Changing the locks is changing the enzyme, so therefore it's denaturing. The enzyme is changing the shape, so therefore your key doesn't fit the lock, and therefore you don't get a reaction happening. So denaturation is where the shape is affected. Changing of the shape is called denaturing, is where the bonds that create the tertiary structure are broken. Now remember, the tertiary structure is made from the Z side chains in a molecule. So what we can do, you can imagine if we change the way that the Z side chains interact, and if we can try and affect those really weak bonds which are holding those tertiary structures together, because remember the types of bonds holding the tertiary structure together are just ionic interactions, very weak ionic interactions, hydrogen bonds which are quite weak. We've got dispersion forces acting in there as well, and they're extremely weak bonds. So if we affect those tertiary structure bonds, the things in the side chains, what we can do is effectively unravel and change the shape of this enzyme, and that will change the enzyme shape, therefore affecting the active site. As it says here, the enzyme gets unraveled and then coagulates into a random shape. So what we're going to do is unravel our enzyme, and then it will change the shape and go into a random shape that no longer has the active site. No longer having the active site means that the reaction will no longer be catalyzed, so therefore the enzyme will effectively stop working. Two ways we can screw with the tertiary structure, two ways we can change the bonds between the Z side chains is one is increasing the temperature. Increasing the temperature basically gives too much energy to these molecules, these atoms, and they start to come apart. So what happens is it starts to get unraveled, and that will change the shape, that will unravel our enzyme and then coagulate it when it cools down into a random shape. The second is changing the pH. If you change the excuse me. The second is changing the pH. If you change the pH, what you do is effectively change the charge on some of these side chains. Changing the pH, putting it into a basic region, will add, it will take away some of the hydrogen, so it will change the pH, will change the acidic side chains, and it will change the areas where we have the NH groups as well. So it'll change the basic side chains, it'll change the acidic side chains as well. So if you change the pH, you're going to stuff up the overall shape and denature your enzyme. Interesting point here, it's only increasing the temperature which will actually denature the enzyme. Decreasing the temperature will make it frozen, and therefore it won't, it can affect the shape in a small term, but then when you increase the temperature again back to the normal time, it hasn't actually broken the bonds which were there. So normally it's just increasing temperature will denature it. And a nice example of how this works is yeast is an enzyme which catalyzes the fermentation of glucose into ethanol and carbon dioxide. What I do, I make a lot of bread at home, so I use a lot of yeast to do that, and I keep my yeast in the freezer. This doesn't kill the enzyme, this doesn't unravel the enzyme. So decreasing the temperature doesn't affect the ability for an enzyme to catalyze a reaction. It will make the reaction go slower, and it can stop it after a while, but when you heat it back up again, it will go back to the normal, the way it works before. So increasing the temperature actually kills and denatures the enzyme. It kills the reaction. It makes it no longer happen, basically. So that's enzyme denaturing. So you need to know that enzyme denaturing screws with this shape here. It stops it from working because it removes the active site. It makes the active site no longer there. A denaturing of an enzyme will change the active site, and therefore the substrate will no longer fit it, and therefore no longer be catalyzed in the breakdown or the formation of your reaction. Moving on to how we can use proteins as medicines, and this is the last part here, so this is our final slide that we're going to do. We've got a summary slide after this, but I probably won't do that because it just summarizes what we need to do. So, medicines. In medicines, what we use proteins for is to look for problems that might be happening. As we said, hormones are proteins. Hormones and other messenger proteins are released when a person is sick. So if your body gets ill, or if your body is under some kind of stress, what your body is going to try and do is counteract that. And to counteract that, it needs to send a message to the parts of your body which are going to fight it off. And the way that they're going to send messages is using hormones. By monitoring these hormones and these proteins that your body sends out in relation to your sickness, we can observe small changes in the body and predict what might become a problem. Because even if we can't see that someone's sick, what their body might be doing is trying to fight it off. And in doing so, they're increasing the hormonal activity, so therefore we can look for these proteins and see when someone's getting sick. 3 examples of these are heart attacks. Obviously, if someone's having a mild heart attack or someone's about to have a heart attack, the heart's going to be trying to talk to other parts of the body, and they're going to release these cardiac enzymes, these heart enzymes. So what we can do is monitor these enzymes or monitor these proteins that the body's sending out and look and see if someone might be under stress. The second one is prostate cancer, and we can actually test for prostate cancer by looking for a certain protein which is given out. I can't remember what PSA stands for, but the PSA is something to do with prostate something something protein. And what it is basically a marker for that disease, for prostate cancer. If you want, Google PSA protein and it'll come up with what it is. But that is one way that proteins can be used to diagnose these different things. Another thing is fertility. Different types of hormones, obviously, in a lady's cycle come and go. They peak at different areas. And one of the fertility hormones in particular is progesterone. And that is a hormone which basically starts off starts off a lady's cycle. So if you have varying amounts of progesterone, what can happen is we can monitor that and see, and we can then try and counteract that by either giving someone this protein or looking at if there's too much, what we can do about that as well. So by monitoring the amounts of proteins or the amounts of hormones and enzymes in a person's body, we can actually use that and get diagnostic data on a person and seeing if they're going to be sick or if they're going to be healthy, or if we can help them make other little people. So that is the last part on proteins. Hopefully this has made sense to you. This is just what the summary will look like. It looks like what you need to know for fats, what you need to know for sugars, what you need to know for proteins. Obviously, the majority of this biomolecule stuff is on proteins, so spend a fair bit of time there, and a little bit's on sugars as well. So you need to know those hydrolysis and all that type of stuff. I'll cover this summary in a bit more detail, probably in an audio podcast. So in that audio podcast, I'll reference these videos and you can understand all about that. But that's it. I hope you've enjoyed this series of biomolecules. And after this, we're going to look at DNA. So take care and stay tuned for the DNA section.